Okay, I think we can now start. I'd like to begin by acknowledging the traditional owner of the land we are living. In my case, I live in Randwick, is the Gadigal and Badigal people. And I would like to pay my respect to elders past and present. Our guest today is Prof. Anne-Florence Bitbol from EPFL. Anne-Florence is a renowned scholar in biophysics. She got her PhD from the Complex Matter and Complex Systems Lab at the University of Paris Diderot. She was a researcher at Princeton University as well as a CNRS in Paris. And she's currently the head of the Laboratory of Computational Biology and Theoretical Biophysics at EPFL Lausanne. And today she will tell us about the impact of population spatial structure on mutant fixation probability. And without any further ado, please welcome Professor Bitbol.
Okay, so thank you very much, Emanuele, for the great introduction. And I would like to thank the organizers for inviting me to give this talk. It's quite exciting to be able to present in Australia and New Zealand. And somehow, I'm kind of, it's kind of funny for me to have this December 1st slide, because actually I'm still on November 30th. So, I'm visiting from last month in a way. So it's kind of funny. Anyway, so so my lab is called the Laboratory of Computational Biology and Theoretical Biophysics at EPFL in Lausanne, Switzerland. And I'm going to tell you about a work on evolution of microbial populations, but very much modeling of this evolution. And in fact, my lab works on evolution at two different scales. We work on the evolution of protein sequences first, where we aim to understand how optimization on the one hand and phylogeny on the other hand shape protein sequences. So that's our molecular scale project. And at the population scale, we aim to assess the predictability of evolution in spatially structured populations. And so today I'm going to tell you rather about the second part, but just to tell you that we are also interested in sequences. Okay, and so my talk today is going to have two parts. First, I'm going to tell you about a general model for spatially structured populations, but it's a bit of an abstract model on graphs. And second, I'm going to tell you about a more concrete case, which is the case of bacteria evolving in the gut. Okay, so let me first start with this model of populations evolving in spatial structures represented by graphs. This is joint work with my former PhD student, Loïc Marek, and my former master's student, Irene Lamberti. So the question that we want to ask is about the impact of spatial structure on the evolution of microbial populations. And this is an important question because natural populations are not usually well mixed. So here I represent this experimental setup of a beaker, which is well agitated, and you may imagine a population of bacteria that evolves in this beaker. In this case, it's more or less well mixed in the sense that all bacteria are competing with all the other bacteria in the beaker. There are some subtleties even in this case, but that's an example of what we can think of as being a well-mixed system. However, in natural populations, very often there is subdivision among different locations. For instance, in an infection, you have subdivision among the organs of a patient. And in an epidemic, for instance, if you think of some virus, you would have subdivision among the hosts that transmit this virus or bacteria. I mean, it's the same idea. Even if you think of another lab setup, which is not this beaker, but a Petri dish, because of the two-dimensional shape of the Petri dish, you would have more competition between the bacteria that are close to one another than with the more distant bacteria, which means that we cannot consider that all bacteria are in equal competition with one another. And this part is, it's quite interesting because most models of evolving populations are done with the assumption that competition is homogeneous. And having non-homogeneous levels of competition is something that complicates the theoretical description. So models with spatial structure have been developed for a long time already, and I will not explain the history, I mean, the rich history of this question, I will just mention one model which was developed starting from this seminal paper in 2005, which is a model that aims to describe general population structures using graphs. And so the idea is that we want to model a complex population structure. And for this, we are going to use a graph, which can be any kind of graph. And at each node of the graph, we are going to assume that there is one individual. And then there are replacement probabilities that are specified on the edges of the graph. That is to say, for instance, here, the individual at vertex 1, at node 1, can replace the individual at node 2 with a probability proportional to W12. Okay. So in order to define the evolution on this graph, we need to define what is called an update rule. So what is an update rule? The idea is that here we have this graph with a fixed number of nodes, and we assume that there is one individual per node in this graph at all times. And this means that each division and death of individuals should result in no variation of the population size. Because of this, when we have one individual that divides, another individual has to die. And it leads to some possible update rules, such as the birth-death or the death-birth update rules. And so let me explain a bit what it means. So the birth-death update rule is an update rule where you first choose which individual is going to divide. So here, in this case, with 3 nodes, you choose one among the 3, and it's this one here, and it is chosen proportionally to its fitness. So fitness, is going to be, it's going to matter at birth, not at death. So that's the choice of the model. We could also put fitness at death, but here I'm going to assume that it's only at division that fitness matters. So different types of organisms are going to have different birth rates, if you will, right? Okay, so the dividing individual is chosen proportionally to fitness, with fitness Fi here. And then when it's chosen, it can replace one of its neighbors on the graph. So the idea is that it divides in 2, and then the offspring replaces one of the neighbors. This is the neighbor that is replaced is chosen proportionally to this quantity Wij, which is in this matrix here. Okay, and so then after this particular update, we have 2 orange individuals, while initially we had only 1. The colors represent here different types, which may have different fitnesses. And that's how the composition of the population is going to evolve. You may make alternative choices. For instance, here I'm showing this death-birth case, in which case, instead of first choosing the individual that divides, we first choose the individual that dies. And so here you may choose this J to die. And because there is no difference of fitness at death, the choice is made completely uniformly. And so here it's the probability to choose J is 1/n, where n is the size, here n is 3. And then once you have chosen this individual that is dying, you have to replace it by the offspring of another one. And so then you choose this other one that is going to divide proportionally to fitness and to the replacement rate Wij. And so in the end here, we have reached exactly the same state as with the birthdays update rule. So at this point, you may think, okay, these are just 2 ways of seeing exactly the same thing. Turns out that it will matter, which is why I wanted to go into some detail of this. And so in this kind of model, what was studied in this paper is the impact of the graph structure on the probability that a mutant is going to take over. So what does it mean? It means that we start from a population where there is one mutant. And so here the mutant is blue and the wild type organisms are orange. And we want to ask what is the probability that after a certain number, a large number of generations, the population is all blue. So in this case, we say that the mutant has taken over or fixed. In fact, there are only 2 alternatives here. If we start from this state where there is one blue mutant and the other are orange, we can either end up with a population that is fully blue or a population that is fully orange. Because of finite size effects, we cannot maintain a mix of the 2 types. If you will, it's a process that is like a diffusion with 2 absorbing boundary conditions. Because once you have reached the elimination either of the mutant or the wild type, then the population can no longer have a composition that changes. It's completely fixed either in mutant or in wild type. Okay, so we want to find the probability that we get only mutants at the end of this evolution. And this property is already well known in the case of a well-mixed population. And if we consider a graph where all the nodes are connected to one another with the same W, then we have this case, which is the well-mixed case, where competition is equivalent between all organisms. However, some structures can amplify or suppress natural selection compared to this well-mixed case. And I'm going to show you just one example, which was described in this paper. It's called the star. And so here you have the structure of the star. There is one node of the graph that is called the center, because it has the central position, and the other ones are called the leaves. And you can see that there are some connections only between the center and the leaves. The leaves are not connected to one another. In addition, there is a strong symmetry. All the leaves are going to be supposed to be equivalent, and in particular to have the same migration probabilities to the center and from the center. So, okay, so that's the star structure. And it was shown that this star, if we are in the birth-death update rule framework, it amplifies natural selection. So what do I mean by amplifying natural selection? Let me show you this plot. So this plot is a simulation of evolution on this star. And here we are showing the probability that the mutant takes over versus the ratio of the fitnesses, mutant to wild-type fitness. So if this ratio Fm over Fw is larger than 1, it means that the mutant is fitter than the wild-type, that it has more chances to divide than the wild-type. Conversely, if Fm is smaller than Fw, it means that the mutant is deleterious, it has less chance to divide than the wild-type. Okay. And what you can see here is in dashed line, the analytical formula for the well-mixed case with 10 individuals. So you can see that the probability of fixation is larger if the fitness of the mutant is larger, which is what you would expect. And now we are going to compare this to the case of the star. So I was telling you that in the birth-death case, the star is an amplifier of selection, and this corresponds to the blue curve. Here, this blue curve is from a simulation of the process. And you can see here that the blue curve is above the dashed curve if the fitness of the mutant is larger than that of the wild type. It means that this excess fitness of the mutant has an effect that is amplified by the fact that the structure is a star. Conversely, if you look at the case where the fitness of the mutant is smaller than that of the wild type, the blue curve is below the dashed curve, which means that this fitness effect is also enhanced by the star. Here, you know, like essentially a beneficial mutant has more chances to fix, and the deleterious mutant has less chances to fix. Okay, so this is, you know, this is quite an interesting property of the star. However, it's a property that depends crucially on the update rule that is chosen. So far, I told you about the birth-death update rule. But here in red, we also have the case of the death-birth update rule. And that, as you can see, the conclusion is completely the opposite. You can see that this curve is very flat and that all fitness differences are reduced and dumped by this death-birth star. And so this is what is called a suppressor of selection. And so we have here these interesting results that the star has a big impact on what mutant is going to take over, or at least on the probability that it takes over. However, this depends on a microscopic dynamics choice. And this is something that is a bit puzzling, especially because natural populations generally don't need to follow such strict update rules. The idea is that in a natural microbial population, bacteria can divide and die without necessarily replacing another bacteria. So there is not a very stringent constraint that you have to maintain the population size exactly fixed. There can be constraints due to nutrient limitation, for instance, but they tend not to be so strict that it's exactly a one-to-one replacement. And this means that having a result that depends on whether the dynamics is death birth or birth rate can be problematic in the general case, even though these dynamics may be relevant in some particular cases. And so we wanted to address this problem by removing the constraint that population size is strictly fixed. And in addition, we thought that because often bacteria compete freely in local neighborhoods, we can probably consider that there are well-mixed populations on each node of the graph instead of having just one individual per node of the graph. So this kind of coarse graining, in fact, was already addressed by this paper. However, in this paper, the fixed size constraint was left. And so we, in addition, removed this fixed size constraint. So in order to lift the fixed population size constraints, what we did is to impose a carrying capacity with a logistic growth term, which means that we have essentially a maximal population size, but that the population size can fluctuate below this value. And so we also chose to stay in the regime where dim sizes, so a dim is a subpopulation, one of these which are going to be on each of our nodes. So we assume that on each of the nodes, the subpopulation or dim has a size that remains close to steady state in this logistic growth framework, so that we don't have any extinction effects, etc. We want to remain close to the previous description, but to lift this fixed size constraint. Okay, and so now we ask the same question as before, which is whether a mutant is going to fix or not in the population. And we also make another hypothesis, which is that we assume that migrations are rare. In particular, we assume that the time between two migration events between two nodes of our graph is going to be longer than the time it takes for a mutant to take over within one of the subpopulations. This means that at each time, we can consider that the population has fixed within each of the dim and that we have just one type at each node. So examples are given below. So you can see, for instance, this graph, which is the clique, where every node is connected to another and they are all equivalent. But at each node, you have a subpopulation, which is well mixed, and which contains only one type of individuals, either orange or blue in this example. And that's because, you know, for the regime of rare migrations, we can consider that at most times, this is true, we have only one type per node, and that upon each migration event, this is going to be the correct way of describing the population. And the advantage of this limit of rare migrations is that then we can describe the state of the system and its dynamics. I mean, we can describe the state of the system by just whether each node is mutant or wild type, and we can describe the dynamics of the system as a Markov chain. So it really simplifies a lot the theoretical description. And we are going to start from the case where you have one dim that is fully mutant. So for instance, here the mutant is blue, and we consider that one of these subpopulations is fully blue, while the others are fully orange, wild type. And the reason for that is that we want to address what happens to the fixation of one mutant. However, one mutant is going to first have to take over in its local population before it can spread, because we are in this rare migration regime. And the first phase, taking over in the well-mixed population, is well known already, so we want to focus on this spreading step. Okay, and so below, I'm showing 3 examples of graphs, which are graphs with very strong symmetries, and these are the ones that we are going to study in more detail. The first one is the clique, where every node is connected to each other, and they are connected with the same migration rate. But then we will move on to these other 2 structures. So first, let me tell you about the results we got from the clique. And so here you can see plots of the fixation probability of the mutant versus the fitness of the mutant. And here the wild type fitness is equal to 1, which means that if mutant fitness is above 1, we have this beneficial mutant that is fitter than the wild type. If it's smaller than 1, we have a deleterious mutant. And we are making the comparison between this clique and a well-mixed system of the same total size. And as you can see here, the results are not very exciting. We find that the clique is behaving almost exactly similarly to the well-mixed system. Okay, so that's, you know, like, in a way, here, structure has no impact. And in fact, it's something that we can show analytically. So I should have said here, the markers are simulations of the process done using a Gillespie algorithm, while the lines are analytical calculations. And these analytical calculations are done using the Markov chain formalism. And you can see here the results of these calculations, where the probability of fixation in the click is given by this formula 1 minus gamma divided by 1 minus gamma to the power d. Here, d is the number of DIMs, which is 5 in these simulation results here. And gamma is the ratio, essentially, of the fixation probability of a wild type in one DIM to the fixation probability of a mutant in one DIM, if all the other individuals are of the other type. There is also this n, which is the equilibrium size, whether you are wild type or mutant, but in practice, this ratio of n is close to 1. So what matters is really this ratio of the fixation probabilities within one DIM. Okay, and so we find this formula, which is very close to the well-mixed case. And we also studied this other structure, which is the cycle. In the cycle, you have connections only along these edges of the graph. And here we can have migrations that are either clockwise or anti-clockwise. They may have different rates. So that's an extra degree of freedom compared to this case where all migrations were equal. And so we have this migration rate asymmetry, which is the ratio of anti-clockwise to clockwise migration rates. And in our simulations, we varied this value alpha. And we can see that it had no impact essentially on the results. And in fact, our analytical calculation based on Markov chains shows that for the cycle, whatever the asymmetry alpha, we have the same fixation probability, which is in fact the same as for the click. So far, we have no impact of population structure on fixation probabilities. However, we can now ask what happens to the star, because the star was found to be an amplifier or a suppressor of selection in the model with one individual per node. So let's look at the star. And here you can see that for the star, results are more interesting. So you can see that in particular, here, they depend on this parameter alpha, which is, again, an asymmetry of migration rates, except that this time, it's an asymmetry between the incoming migration to the center, which is MI, and the outgoing migration from the center to the leaves, which is MO. So this migration rate asymmetry has a big impact on the results. And you can see here that the simulation results match the analytical ones quite well. And in the analytics, again, based on our Markov chain formalism, we can find the fixation probability for the star. And it has a more complicated form than before. But what is interesting is that it still depends on number of DIMs D. So here it's 5 little DIMs. And the ratio gamma of the fixation probabilities of one wild type in one DIM to one mutant in one DIM. But in addition to that, it also depends on alpha, our migration rate asymmetry. And so that's reflected in these curves. And you can see that if alpha is small, then we have essentially a suppressor of selection. Having a nice fitness for the mutants is not helping as much as in the well-mixed population. While if alpha is large, we seem to have a very slight amplification effect. Because in the model with one individual per DIM, it was known that amplification in the star was larger for larger numbers of nodes in the graph. We then moved on to a case with the number of nodes, where the number of nodes is larger. So we took D equal to 100. And here we are showing the corresponding analytical predictions. And you can see that indeed here, for large values of alpha, we have a larger amplification of selection, which is consistent with what was found in the model with one individual per node. Except that here, the conclusion whether the star is an amplifier or a suppressor of selection depends crucially on the migration asymmetry alpha. If alpha is 1, by the way, you can see that we recover the results from the well-mixed population. In fact, technically, we recover exactly the ones from the click that I showed before. So this asymmetry of migration really enables one to tune the star from being an amplifier to being a suppressor of selection. Okay, so now what can we say about the comparison between this model and the model with one individual per node that I introduced and that was first introduced in this paper? And so we made a mapping between these two models. the idea is that our model is essentially a coarse graining of this seminal model. So we consider the mapping by saying that a birth-death step in this model with one individual per node is equivalent to a migration plus fixation step in our model. Essentially, in our model, to change the state of a dim, we need to have a migration that goes to this dim, and then the mutant needs to fix in this new dim where it has migrated to. So that's what replaces, what plays the role of the birth-death step in the model with one individual per node. And so we can construct a formal mapping where the fitness is replaced by the fixation probability times the equilibrium size, and the replacement probability is replaced by the migration rate. And then the formulas are equivalent. However, we need to impose one extra constraint, which is that in our model, there is no constraint of fixed size. This is a crucial difference with the model with one individual per node. And because of this, in our model, there is no requirement that the sum of the probabilities of replacement has to be 2, which is here a sum over j, meaning that the sum over all destinations of one individual that divides has to be 1. Essentially, the dividing individual has to go somewhere, right? So summing over destinations has to be 1. And so in our model, we don't have this constraint. So now let's impose this constraint to our model. And if we do this, we find that in the case of the star, we recover exactly the formula that was found for the birth-death case in this model with one individual per node of the graph. And in this case, the number of dims minus 1 plays the role of the migration rate asymmetry alpha. And so it means that we can also look at what happens versus migration asymmetry and number of dims, and plot the case of the birth-death model in the case with one individual per node, which would be here this white curve, where alpha is equal to d minus 1. So here, this plot is showing you a heat map of the ratio of fixation probability in the star versus the well-mixed case, which means that if this ratio is larger than 1, if we are in blue, we have an amplifier of selection, while if we are in red, we have a suppressor of selection. And we are showing this ratio versus migration asymmetry and number of dims, which are independent in our model, but which were constrained to be related by alpha equal to d minus 1 in the birth-death regime of the model with one individual per node. So what you can see is that in our model, we have vastly different results depending on migration asymmetry. We switch from being an amplifier to being a suppressor, while if you are constrained to be on this line, you see the star as being just an amplifier of selection. So this means that our model has this extra parameter, which is migration rate asymmetry, and which determines whether the star is going to be an amplifier or a suppressor. And what is interesting is that this parameter is actually a parameter that can be tuned experimentally. If you consider an experimental system which comprises populations, subpopulations, which have migrations between them, then it's not obvious to say that they evolve in the death-birth or in the birth-death way. But experimentally, you can tune migration asymmetry. For instance, if you can pipette some fraction of the population of one dim and re-inject it in the other, then you will control migration rate asymmetry. So it's something that opens to comparison to experiments, which is all the more interesting that such experiments are being done actually currently, for instance, in the group of Wisconsin. So to summarize on this part of my talk, we have developed a coarse-grained model of populations that are subdivided on graphs, which lifts the fixed size constraints and which makes migration, birth, and death independent. And this recovers some of the results of the previous model, but it also elucidates the apparent paradox in the constant size model that amplification of suppression depends on the update rule, which is a microscopic choice. Here, instead, the fact that we have an amplifier or a suppressor of selection depends on migration rate asymmetry, which is something that can be tuned experimentally. And so far, I have shown you on these quite simple graphs, and it's interesting to look at more complex graphs, and also to address the case where we have dims of different sizes. So we have done a simple example of this with one large and one small dim. And also, an important question would be to go beyond rare migrations, because this was a very a simplifying hypothesis that we made for the calculations to be tractable, essentially, but it's very interesting to go beyond it. And also to study fixation times in addition to fixation priorities. And I think another interesting perspective is to look at the impact on concrete situations of this subdivision, for instance, the impact of structure on antibiotic resistance evolution. Okay, so that's it for my first part. If you're interested to know more, we have a reference about this. And I particularly thank my two students, Loïc Marek and Irene Lamberti, for their great work on this project. So Loïc is now a postdoc in Bern, and Irene is a PhD student at EPFL in another group. And I also thank the ERC for funding. And I'm happy to take questions on this part if there are any, but I mean, of course, if you prefer to wait until the end, I'm also happy. I'm happy either way. So, just want to say that if there are any pressing questions, I can take them now.
Any question?
Can I ask one, Manu?
Yeah, sure. I also want to ask one. So go ahead, Richard.
So, actually, Anna, I was thinking. I mean, when you presented the clique versus the cycle, and showed that there's not much difference between in terms of the fixation probabilities. Presumably, there is a difference between the mean first passage time.
Yeah, you're right. Indeed, yes.
So this is a sort of, in some sense, there's a metric choice here as to where you see spatial structure.
Yes, you are completely right. So that's a very good remark. The fixation priority and fixation time are two very important quantities in this process. And here it's in terms of the priority that they behave the same. In fact, there is a theorem, which is called the circulation theorem in the case of one individual per node, which states that if all the incoming migration, the sum of incoming migration and the sum of outgoing are the same for each node, then fixation priority is the same as in the well-mixed case. Okay. Yeah, but that's only for probability. You are completely right that for the time, it's different. And indeed here, it would be different. Yes. So that's a very good point. And the reason why we didn't focus on time here is because we are in the rare migration regime, which means that fixation times are slow anyway. We thought that it would be better to first address more frequent migrations before looking at time again. But we can obtain time also using this Markov chain framework here.
Yeah, no, sure. I just was just to confirm my intuition, making sure I actually understood.
But that's completely correct. Yes.
Yeah. Thank you.
Thanks.
All right. And my question would be, can you name some examples of real biological systems that correspond to the cliche, the cycle, or the star?
Yeah, look, so that's a great question too. I mean, these models are quite abstract. They are nice because they are highly symmetric, which means that we can make calculations on them rather easily.
Yeah.
Okay, so that's theoretical
information.
I mean, however, the idea of the clique is a population where all subpopulations are related to one another equivalently. So it's a case where you have no notion of distance, that will impede migration or stuff like this. If you were to consider kind of 2D network, it would model, it could model, for instance, an epithelium, which is 2D, stuff like this. The star has an interesting property, which is that it can help us to understand more complex networks that have hubs, right? So the star per se seems to be a bit artificial. I mean, like you need to have one center connected to leaves. But if you consider a random network that has lots of hubs, for instance, then the star becomes relevant because the center is essentially a hub. So that would be, I would say studying more realistic networks, such as random networks of different kinds, would be very interesting. And it has been done in the models of one individual per node. The star is interesting because it allows us to understand what happens in the case of one hub. Okay. You can also think of organs in a patient where, like some organs are more central than others, notion of center and periphery. Right?
Yeah. Right. Thanks. I think most people will ask questions at the end. So
okay, sure, no problem. I just wanted to open this possibility, but
thanks for that. It was a really clear explanation.
Thank you very much. So, okay, let me move on to my second part now. So the second part is going to be a more concrete case of spatial structure. So we are going to talk about the fate of bacterial mutants in the gut, and specifically focusing on neutral bacterial mutants for now. This is joint work with my close collaborator, Claude Loverdeau from Paris, Sorbonne University, and with our postdoc, Darka Labevich. Okay, so we are going to talk this time about gut microbiota. And gut microbiota are very interesting, first of all, because they are abundant. There are as many bacteria as human cells in the human body, and most of them are in the gut, mainly in the colon. There is also a high diversity of species in the colon, and they can have a big impact on health, which I'm not going to discuss here, but they are also important for medical reasons. And one source of this diversity of species is evolution within the gut. We are going to focus on this part. And in fact, it can have public health implications because the gut can be a reservoir of drug resistance, both in humans and in animals. So understanding how drug resistance would evolve in the gut can be of importance for this. And so in order to study this, we are going to view the gut as an environment for bacterial evolution. And we are going to take into account some key points of the gut, which is that essentially the gut is a cylinder and there is flow along one direction, along the main axis of the gut. And most bacteria are actually carried with this flow, with the digester. Some of them are actually associated with the mucus and stay close to the edges of the gut. We are not going to focus on them. We are going to focus on the majority, which are carried with the digester. In addition to this overall flow, there are also contractions of the walls of the gut, which help with the mixing of the digester. So it helps the digester being well mixed. And these contractions are quite hard to model. I mean, you would imagine that you need to have boundary conditions that move, etc. However, it was shown in a recent model that also made comparisons to some experiments that to some good approximation on a large scale, these contractions can be modeled as having the effect of a large effective diffusion coefficient. So that's the model we are going to take, that in the gut there is essentially a directional flow towards the outside and an effective diffusion that is quite large. And the boundary conditions are going to be taken this way. So there is an inflow of food at the entrance, but no inflow of bacteria. This is because we focus on the entrance of the colon, and the concentrations of bacteria are much lower in the small intestine than in the colon. So we consider that there is essentially no inflow of bacteria. And then at the end of the slice of colon that we consider that there is free outflow to the rest of the colon. So these are our boundary conditions. And this system is going to give rise to strong gradients of food and bacterial concentrations. And qualitatively, you can imagine that at the entrance, there is a lot of food that enters. Then this food is being consumed and converted to bacteria. So concentration of food decreases, concentration of bacteria increases. So this system, which is very simplified, a very simplified model of the gut, a minimal model of the gut, is already quite complex in the sense that it has this minimal hydrodynamics of directional flow and diffusion, and also these strong gradients. And we want to ask, what is the impact of this spatial structure on the evolution of gut bacteria? In order to address this question, so we considered this model that in fact I already described, and we describe this model in terms of partial differential equations, which is something that was introduced in this paper. So we base our model on the one of this paper, where you have a differential equation for the food concentration and another one for the bacterial concentration. These differential equations, these partial differential equations are very similar to one another. The idea is that both of them have a diffusion term modeling the contractions and a velocity term modeling the overall flow. And then there is a term that converts food into bacteria. So this term is nonlinear, and it reflects the fact that there is a saturation of the growth rate, which is incorporated in this mono constant. And the factor alpha, which is different between these two equations, is the yield of the food to bacteria conversion. So here, essentially, we have this function that converts food into bacteria. Food is being eaten by the bacteria. Okay, so these are the equations. And the boundary conditions, which I mentioned before, are given here. We have this inflow of food, no inflow of bacteria, and then free outflow at the other edge. Okay, so this is a mathematical description, and I'm going to show you the stationary state of this system in order to have an idea of what happens, and then we are going to discuss what happens to mutants. So for now, let's imagine that there is only one species of bacteria. We only have food and bacteria, so two fields to describe. And here we are showing the stationary profiles of food and bacteria in a numerical resolution of these coupled partial differential equations. So you can see here the food concentration, which is in green. It essentially decreases and drops as you go along the main axis of the gut. And simultaneously, the concentration of bacteria increases and saturates. And that's because bacteria are eating the food, and then at some point they, it saturates, all the food is being eaten, and they cannot grow anymore. So that's the idea. And we have also plotted here in red the local reproduction rate, which is given by this function. Essentially, it's proportional to food, but there is a saturation. Okay. So local reproduction rate is also something that decreases quite steeply, and after some position in the gut, there is no more reproduction because there is no more food to reproduce. Okay, so that's the stationary state. But of course, this is plotted for a given choice of diffusion coefficient, velocity, and also the other parameters. And we may ask how generic these very steep dependencies of the profiles are when we vary velocity and diffusion values. In order to address this, we are going to quantify the spatial dependence by the difference of food at the entrance minus food at the exit divided by the inflow of food, with the idea that if this value is close to 1, it means that we are in this very spatial case, while if this value is close to 0, it means that there is almost no variation of food concentration between the entrance and the exit. Okay, so that's our quantity, and we are going to plot it for different values of V and D. So here is the heat map when we vary velocity and diffusion coefficients. And we are plotting this spatialness quantity here, which is red if it's close to 1, and purple if it's close to 0. And what we can see is that there are regimes where the profile is essentially not spatial, so purple here, especially if the velocity is too large. Then what happens is that all the bacteria are essentially flowing out of the system. So these are in fact these limits, which are the lines here. So they correspond to the case where we have this everything flowing out of the system. And there are, oops, sorry, there, oh, okay. Here we are. So there are also some cases where the profiles are very flat, but without everything flowing out. And these are these purple cases here for low velocity. Okay, so in between, we have this interesting behavior where the profiles are more spatial. And you can see here these regimes, and the cyan dot corresponds to what is plotted in this curve. So it's this choice of parameter that we made here. And we think that this is the one that is relevant for the gut, I mean, this strongly spatial regime at least, because in the gut, there are strong gradients of concentration of bacteria and food. Okay, so that was to mention that the behavior of the system depends on the parameter quite strongly. And now we want to ask what happens to mutants in this system. So we have this stationary state, with bacteria that have a certain profile, etc. And now imagine that we add mutants somewhere, so they appear spontaneously at some location. What is going to happen to them? Okay. So the first thing that we look at is the mutant to wild type ratio, M over B. Versus the introduction position of the mutant, so the initial location where we place the mutant. And this ratio, when we look at it using these numerical resolution of our partial differential equation, it has this shape which decays with xm. This is qualitatively quite natural because if a mutant appears close to the entrance, it has more time to divide before it's washed away from the system by the flow than a mutant that appears at the end. Okay, so that makes sense. And we also plotted here in pink this number of reproduction per unit volume and unit time, which is essentially this function, proportional to the local reproduction rate times the number of bacteria. And what we can see here is that this number of reproduction has a maximum at an intermediate position. This is because if you are close to the entrance, there is a lot of food, but there are few bacteria. And if you are close to the exit, there are a lot of bacteria, but there is no more food left. So that's why you have an intermediate maximum. And this is quite interesting because, in fact, if mutants appear upon division, for instance, due to a replication error, then you would have a probability of appearance of the mutant that would be proportional to this number R and to this pink curve. So mutants would be more likely to appear here, but on the other hand, they would take a larger proportion if they appear here. And so in order to look at the fate of the mutant, we are going to consider this ratio M/B as the fixation probability of neutral mutants. And this is because here we are in a deterministic description with these partial differential equations. But if there was any stochasticity, any finite size effects, we would have fixation of one type or the other, as in the first part of my talk. And for neutral mutant, fixation probability is given by the ratio of initial number of mutants to total number of bacteria. And if M is smaller than B, then it's approximately equal to M/B. So this ratio is going to give the fixation probability of the neutral mutants. And we are going to consider the product of this probability of fixation times the probability of appearance of the mutant at a given location in order to look at where the mutants that fix are more likely to have appear. And we can see that this product also has a maximum at an intermediate place. So this is the location where it's more likely to find a mutant that is going to take over. Okay, so far this was all describing what happens to mutants that appear at certain places. But now we also want to understand what happens overall if mutants can appear anywhere in the gut. What is their overall fixation probability? So we average over the place where they appear by taking the fact that they appear proportionally to this reproduction number, R, and then fixation probability is M/B. So this is our overall fixation probability. And we want to ask how it is impacted by the spatial dependencies in the gut, which we can quantify by this number, so food at the entrance minus food at the exit divided by incoming food. And we want to compare this to the well-mixed case, where the fixation probability would be simply the ratio of mutants number introduced divided by total number of bacteria in the system. Okay, so let me show you the results from our numerical resolution. You can see here fixation probability versus total population. And this cyan line corresponds to the expectation in the well-mixed case. It's NM/NT. And the markers correspond to the results in our simple model of the gut. And you can see here that we have a strong deviation from the line in some regime of the parameters. So let's go a bit deeper into this. You can see that here we have colored the markers with a color ranging from purple to red. And in fact, it corresponds to this quantity that quantifies the spatialness of the profile. What we see here is that the deviation from the well-mixed expectation is strong when we have very strongly spatial systems, which are in red here. Okay, essentially, when profiles are almost flat, we have something that is in good agreement with the well-mixed expectation, but then it deviates, and then it comes back to the well-mixed expectation. So this is obtained by, in fact, scanning along one horizontal line, and the markers correspond to this horizontal line. For instance, this black circle corresponds to the case where we scan along this line. The full circle is this curve. Okay, so how can we make sense of this? We introduced a notion of active population in order to make sense of this. The idea is that in this system, we have a region of the gut where almost no division can take place because there is no more food. That's the right-hand side region. And so it means that only the part of the population that is on the left is active in the sense that it is reproducing. Here, in this case, where we show again the profile of bacteria and the local reproduction rate versus position in the gut, the active population, which is shown in gray here, is defined as the population such that the reproduction rate is equal at least to half of its maximum value. And okay, so that's how we define the active population. And the idea is that this active population is the population that will matter for fixation, because the rest, is inactive and it's just dormant bacteria, essentially. Okay, so now we plot fixation probability versus the active population size. And what we can see is that this time, the results fall very nicely on this line, which is the well-mixed expectation, but the well-mixed expectation for a population of the size of the active population. While before we had plotted the case of a population of the size of the total population. So this shows that the active population is really what matters for our system, and that it explains the fixation probability that we observe that are higher than in a well-mixed system. So to conclude on this, the gut features some strong spatial dependencies of food and bacterial concentration. And this spatial structure of the gut is interesting because it favors the spread of neutral mutants and the evolution of the bacterial population composition. So in a way, it favors diversity, right? Probability that the mutant takes over, a neutral mutant, is higher in the gut than in this well-mixed system. So far, we have only considered the case of the neutral mutants because it's formally simpler. It would be interesting to consider beneficial and deleterious mutants and to see if the gut is an amplifier or a suppressor of selection in the previous sense. And also, our model of the gut is really a minimal model. It would be interesting to make it more realistic, and also to include both the mucus and the lumen, because here we focus just on the digester, not on the mucus, which can also be very interesting. So with this, I would like to thank my close collaborator, Claude Lauverdeau from Laboratoire Jean Perrin at CNRS and Sorbonne University in Paris, which is actually my former institution, and our joint postdoc, Darka Lavavich. And if you'd like to know more, we have a preprint about this work. And I also thank Sorbonne University for funding by an emergence grant, which funded 1 year of Darka's postdoc, where she did great work on this project. And I would like to end this by thanking also my current group members, some of whom are working on closely related topics, and my current funding sources, which are EPFL and ERC. And with this, I would like to thank you very much for your attention, and I'm very happy to take any questions.